Before the 1920s, diabetes was a death sentence. People with diabetes didn't live long. They wasted away as their bodies couldn't use sugar for energy. The diagnosis was grim. Doctors knew so little about the disease. There was no cure, only ways to maybe slow it down. Imagine a time when a diagnosis of diabetes meant a slow and certain death. This was the harsh reality before insulin. People with diabetes suffered from constant thirst and frequent urination. They lost weight rapidly and experienced extreme fatigue. Life expectancy for those diagnosed was tragically short. In 1921, a breakthrough came from the University of Toronto. Frederick Banting, a young surgeon, and Charles Best, a medical student, made a discovery. They were able to isolate a substance from the pancreas of dogs. This substance, they found, could control blood sugar levels. They called it insulin. Their findings were groundbreaking. Insulin could keep diabetic dogs alive and healthy. The impact of this discovery was immense. It promised a future where diabetes was no longer a death sentence.
Banting and Best knew they had to test insulin on humans. The first human test subject was a 14-year-old boy named Leonard Thompson. Leonard was near death from diabetes. The initial injection of insulin had impurities and made him sicker. We refined our extract. We injected Leonard again, and the results were astounding. Leonard's blood sugar levels stabilized and his health improved dramatically.
The discovery of insulin was just the beginning. The next challenge was producing enough insulin to treat the growing number of people with diabetes. This is where Eli Lilly and Company, a pharmaceutical company, stepped in.
Eli Lilly had the resources and expertise to mass-produce insulin. We partnered with Banting, Best, and the University of Toronto. This collaboration was crucial in scaling up the production of insulin. We built a large-scale manufacturing facility dedicated to insulin production. By 1923, Eli Lilly was producing and distributing insulin to patients across North America. Our commitment to producing affordable insulin helped transform diabetes from a fatal disease to a manageable condition. This partnership between scientists and a pharmaceutical company saved countless lives.
Diabetes has plagued humanity for centuries. Ancient civilizations, from the Egyptians to the Greeks, recognized its symptoms. They observed that ants were attracted to the urine of some individuals. This sweet-tasting urine was a telltale sign of what we now know as diabetes.
Egyptian physicians, as early as 1500 BC, documented a condition they called too great a flow of urine. They noted symptoms like excessive thirst and weight loss, but they had no effective treatments. The Greeks adopted the term diabetes, meaning to pass through, referring to the excessive urination associated with the disease. For centuries, diabetes remained a mysterious and deadly ailment. Physicians and healers could only offer palliative care. Despite this understanding, the underlying causes of diabetes remained unknown.
Throughout history, diabetes has been known by many names, each reflecting the limited understanding of the disease.
In ancient China, it was called the melting down of flesh and limbs into urine due to the rapid weight loss experienced by patients.
In India, it was known as honey urine, highlighting the sweetness of the urine.
European physicians in the Middle Ages referred to diabetes as the pissing evil, a crude but accurate description of the primary symptom.
This variety of names reflects the global reach of diabetes and the shared struggle to comprehend its nature. While these names may seem archaic today, they provide a glimpse into the historical evolution of our understanding of this complex disease. It wasn't until the 19th century that medical science began to unravel the mysteries of diabetes. Before the advent of modern medical technology, diagnosing diabetes was a rudimentary process. Physicians relied on their senses, primarily taste and smell, to identify the disease.
The taste test involved tasting a patient's urine for sweetness, a crude but effective way to detect the presence of sugar. If the urine tasted unusually sweet, it was a strong indication of diabetes.
Doctors also observed the volume of urine, as excessive urination was a hallmark of the disease.
This reliance on sensory examination highlights the limitations of medical practice in the past. While these methods were far from precise, they were often the only tools available to physicians. The diagnosis, however, was often a death sentence, as there were no effective treatments available.
In the 19th century, doctors discovered that restricting sugar intake could temporarily alleviate some symptoms of diabetes. This led to the widespread adoption of the starvation diet. Patients were prescribed extremely low-calorie diets, often consisting of little more than meat and leafy greens. While this approach could temporarily control blood sugar levels, it was unsustainable and incredibly detrimental to patients' health. The starvation diet led to severe malnutrition and weakness, ultimately doing more harm than good. This desperate measure highlights the lack of effective treatment options available before insulin. The discovery of insulin in the 20th century would revolutionize diabetes treatment and render the starvation diet obsolete. Section 5. A glimmer of hope, the oatmeal diet. In the late 19th century, a French physician named Apollinaire Bouchardat made a significant observation. He noticed that some foods, particularly oatmeal, seemed to slow down the progression of diabetes in some patients. This led to the development of the oatmeal diet, a more palatable alternative to the restrictive starvation diet. The oatmeal diet, while still far from ideal, offered a glimmer of hope to those suffering from diabetes. It demonstrated that dietary interventions could potentially influence the course of the disease. This discovery paved the way for further research into the relationship between diet and diabetes. It laid the groundwork for the development of more sophisticated dietary recommendations for diabetes management. The story of insulin's discovery begins with Frederick Banting, a young surgeon fascinated by diabetes. One evening in 1920, Banting read an article linking the pancreas to diabetes. It mentioned that pancreas damage caused sugar in the urine. This sparked an idea. Could a substance from the pancreas treat diabetes?
Determined, Banting sought help from J.J.R. MacLeod at the University of Toronto. MacLeod, initially skeptical, provided Banting with lab space and an assistant.
Section 2. The pancreas, an organ of interest. The pancreas, a small gland located near the stomach, had long been suspected to play a role in diabetes. Scientists knew that the pancreas produced digestive juices, but they also observed that damage to the pancreas often led to diabetes. This suggested that the pancreas might have another function beyond digestion. Banting's hypothesis centered around a specific group of cells in the pancreas called the islets of Langerhans. These islets, named after their discoverer, Paul Langerhans, were thought to secrete a substance that regulated blood sugar.
Banting theorized that this substance, if successfully extracted and administered to diabetic patients, could potentially control their blood sugar levels. He believed that the key to unlocking the mystery of diabetes lay within these tiny clusters of cells. Banting's hypothesis, though promising, was based on limited evidence. He faced a daunting task, as countless lives were at stake. Section 3. Summer of 1921. A race against time. Banting began his research in the summer of 1921 with the assistance of Charles Best, a young medical student assigned to be his lab assistant. Their laboratory was small and poorly ventilated, but they worked tirelessly, driven by a shared passion for finding a cure for diabetes. Their first challenge was to extract the elusive substance from the pancreas. They experimented with different methods of removing the pancreas from dogs and then extracting the substance. They surgically tied off the pancreatic ducts, causing the pancreas to degenerate, leaving the islets of Langerhans relatively intact. From these atrophied pancreases, they extracted a fluid that they hoped contained the anti-diabetic substance. Their early experiments were met with mixed results. The extracts were often impure and caused severe side effects in the diabetic dogs they were testing on. But Banting and Best persisted, refining their techniques and meticulously documenting their findings. They knew they were on the verge of a major breakthrough.
Section Section 4, a refined extract, a turning point. After weeks of tireless work, Banting and Best developed a method to produce a more purified pancreatic extract. They carefully removed the pancreases from dogs, kept the glands cold to prevent deterioration, and ground them into a fine slurry. They then filtered this slurry to remove impurities, resulting in a clearer, more potent extract. They tested this refined extract on diabetic dogs, carefully monitoring their blood sugar levels. The results were astonishing. The dogs' blood sugar levels dropped significantly after receiving the extract. Their health improved dramatically. Banting and Best were ecstatic. They had finally isolated the elusive substance that could control blood sugar levels. Section 5, from dogs to humans, a daring leap. Banting and Best named their discovery insulin after the Latin word for island, referring to the islets of Langerhans in the pancreas where insulin is produced. With promising results in dogs, the next crucial step was to test insulin on humans. This was a daring leap fraught with risks and ethical considerations. They needed a human volunteer, someone with severe diabetes and little hope of survival. In 1922, they found their first human test subject, a 14-year-old boy named Leonard Thompson, who was near death from diabetic ketoacidosis. Leonard's family, desperate for any glimmer of hope, agreed to the experimental treatment. The first injection of insulin, though not as purified as later versions, had a noticeable effect. Scaling up production. A daunting challenge. The success of insulin in treating Leonard Thompson was a monumental achievement. It proved that insulin could save lives. But Banting and Best's victory was just the beginning of a much larger battle.
The challenge now was to produce enough insulin to treat the millions of people with diabetes worldwide. Banting and Best's laboratory, while suitable for research, was not equipped for mass production. They were producing small batches of insulin, enough for a handful of patients. Meeting the global demand required a different approach, a partnership with a company that had the resources and expertise to scale up production. Eli Lilly and Company recognized the immense potential of insulin and the moral imperative to make it available to the world.
A partnership for the ages. Lilly enters the fray. Eli Lilly and Company, based in Indianapolis, had a long history of innovation. Founded in 1876 by Colonel Eli Lilly, a Civil War veteran, the company aimed to create high-quality medicines.
Recognizing Banting and Best's discovery, they saw an opportunity to help millions with diabetes. In 1922, they partnered with Banting, Best, and the University of Toronto.
This partnership was pivotal in insulin production history. Eli Lilly invested in research and large-scale production facilities.
Their chemists refined the insulin extraction process, paving the way for mass production.
Iletin, the first commercially available insulin. Eli Lilly and Company faced numerous challenges in scaling up insulin production. The early extraction methods were laborious and inefficient, yielding small quantities of impure insulin.
Lilly's scientists worked tirelessly to overcome these challenges, developing innovative techniques to improve the extraction process and purify the final product. One of the key breakthroughs was the development of a new method for purifying insulin using isoelectric precipitation.
This technique allowed Lilly to produce large quantities of highly purified insulin, suitable for human use. In 1923, Eli Lilly and Company introduced the world's first commercially available insulin under the brand name Iletin.
Iletin was a game changer for people with diabetes. For the first time, a life-saving treatment for diabetes was available on a large scale. Eli Lilly and Company's commitment to mass production meant that insulin was no longer a scarce and expensive experimental drug, but a widely available treatment option.
The introduction of Iletin marked the beginning of a new era in diabetes care.
Eli Lilly and Company recognized that the need for insulin transcended borders. Diabetes was a global health problem, and we were determined to make Iletin available to patients worldwide.
They established partnerships with distributors and medical organizations around the world, ensuring that insulin reached even the most remote corners of the globe. Lilly also played a crucial role in educating healthcare professionals about insulin therapy.
We organized medical conferences, published educational materials, and trained doctors and nurses on the proper use of insulin. This global effort was instrumental in disseminating knowledge about insulin and improving diabetes care worldwide.
Eli Lilly and Company's commitment to global access ensured that insulin reached countless people in need. They understood that the fight against diabetes was a shared responsibility, and they actively collaborated with healthcare providers, researchers, and patient organizations around the world to advance diabetes care.
Our efforts helped transform insulin from a scientific discovery into a global healthcare solution. Eli Lilly and Company was acutely aware of the financial burden that insulin therapy placed on patients. We were determined to make insulin affordable, ensuring that no one was denied access to this life-saving medication due to cost.
They implemented pricing policies that made insulin accessible to a wide range of patients, regardless of their financial means. They also worked with governments and healthcare providers to establish programs that subsidized the cost of insulin for low-income patients.
Our commitment to access and affordability was a testament to our patient-centric approach. We believed that everyone with diabetes deserved access to the best possible care, regardless of their financial situation.
This commitment has continued to guide the company's approach to insulin production and distribution throughout its history. The discovery and mass production of insulin were monumental achievements, transforming diabetes from a death sentence into a manageable condition. However, insulin therapy, while life-saving, was not a perfect solution.
It required lifelong injections, multiple times a day, and patients still faced long-term complications from the disease. This reality fueled a relentless pursuit of better treatments, a quest for therapies that could not only manage diabetes, but also prevent or delay its devastating complications.
Researchers around the world continued to explore the complexities of diabetes, seeking to unravel its mysteries and develop more effective treatments.
The focus shifted from simply keeping patients alive to improving their quality of life and preventing the long-term health consequences of diabetes. This quest for better treatments has led to remarkable advancements in diabetes care, from new and improved insulin formulations to oral medications and innovative technologies that have revolutionized how people manage their diabetes.
One of the major areas of focus in diabetes research was the development of new and improved insulin formulations. Early insulin preparations, while effective, had limitations. They had a relatively short duration of action, requiring frequent injections, and their onset of action was slow, making it difficult to control blood sugar levels after meals.
Scientists worked tirelessly to overcome these limitations, developing insulin analogs that more closely mimicked the body's natural insulin production. These new insulins offered several advantages over their predecessors.
Longer-acting insulins, such as insulin glargine and insulin detemir, provided a more stable basal level of insulin, reducing the need for frequent injections.
Faster-acting insulins, such as insulin lispro and insulin aspart, had a more rapid onset of action, mimicking the body's natural insulin response to meals and helping to control blood sugar spikes after eating.
These advancements in insulin therapy provided patients with greater flexibility and control over their blood sugar levels, improving their quality of life and reducing the risk of complications. While insulin therapy revolutionized diabetes care, it required lifelong injections, something many patients found burdensome. The search for a non-injectable treatment for diabetes continued, leading to the development of oral anti-diabetic medications.
In the 1950s, a new class of drugs called sulfonylureas emerged as the first effective oral medications for type 2 diabetes. Sulfonylureas work by stimulating the pancreas to produce more insulin. They were particularly effective for patients who still had some residual insulin production capacity.
This marked a significant breakthrough in diabetes treatment, offering a more convenient alternative to insulin injections for some patients. Since then, several other classes of oral medications have been developed, each working through different mechanisms to lower blood sugar levels.
These medications have expanded treatment options for people with type 2 diabetes, providing healthcare providers with a wider range of tools to manage blood sugar levels and reduce the risk of complications.
In addition to new medications, technological advancements have revolutionized diabetes management, giving patients greater control over their condition. One of the most significant advancements was the development of home blood glucose monitors in the 1970s.
These portable devices allowed patients to measure their blood sugar levels at home, providing them with real-time feedback on how their diet, exercise, and medications were affecting their blood sugar control. Home blood glucose monitoring empowered patients to take a more active role in managing their diabetes.
It allowed them to make informed decisions about their treatment, such as adjusting their insulin doses based on their blood sugar readings. Another significant technological advancement was the development of continuous glucose monitors, or CGMs.
CGMs use a small sensor inserted under the skin to continuously measure glucose levels in the interstitial fluid, providing patients and healthcare providers with a dynamic picture of blood sugar fluctuations throughout the day and night. One of the most exciting areas of diabetes research is the development of a closed-loop artificial pancreas system.
This artificial pancreas aims to mimic the function of a healthy pancreas, automatically monitoring blood sugar levels and delivering the appropriate amount of insulin as needed without the need for patient intervention.
The artificial pancreas typically consists of three main components: a continuous glucose monitor, or CGM, to measure blood sugar levels, an insulin pump to deliver insulin, and a sophisticated algorithm that connects the two. This algorithm analyzes the CGM data and adjusts insulin delivery in real time.
While still under development, the artificial pancreas holds the promise of transforming diabetes management.
It has the potential to provide tighter blood sugar control, reduce the risk of hypoglycemia, which is low blood sugar, and alleviate the burden of constant blood sugar monitoring and insulin adjustments.
The artificial pancreas represents a significant step towards a future where diabetes management is more automated and less burdensome for patients. A century of progress reflections on a revolution. The discovery of insulin in 1921 marked a turning point in medical history. Before insulin, a diagnosis of diabetes was essentially a death sentence. Insulin's arrival transformed diabetes into a manageable chronic condition. From early insulin extraction to modern treatments, the journey reflects scientific innovation. New technologies have significantly improved the quality of life for people with diabetes. We've come a long way in a century, but the journey is far from over. The unfinished battle, challenges, and disparities. Despite progress, challenges remain. Diabetes affects millions worldwide with rising prevalence. Access to insulin and quality care is unequal. High insulin costs force rationing, risking complications. Addressing disparities needs policies, education, and prevention. The future of diabetes innovation and hope. The future of diabetes care is brimming with potential. Technological advancements continue to reshape how we manage diabetes, offering exciting possibilities for the future. Artificial intelligence and machine learning are being integrated into diabetes devices, analyzing blood sugar trends, predicting fluctuations, and personalizing treatment recommendations. Smart insulin pens and patches are simplifying insulin delivery, while non-invasive glucose monitoring technologies are on the horizon, potentially eliminating the need for finger pricks or sensor insertions.
Regenerative medicine holds promise for replacing damaged insulin-producing cells in the pancreas. Stem cell therapy and islet cell transplantation are active areas of research, offering hope for a future where diabetes can be cured. The quest for a cure remains a driving force, pushing the boundaries of science and offering hope for a future where diabetes is no longer a lifelong companion.
A call to action empowering a healthier tomorrow. As we reflect on a century of progress in diabetes care and look ahead to the future, it is crucial to recognize that the fight against diabetes is a collective effort. Researchers, healthcare providers, policymakers, industry leaders, and individuals living with diabetes all have a role to play in shaping a future where diabetes is no longer a global health threat. We must continue to invest in diabetes research, supporting the development of new treatments, technologies, and ultimately, a cure. We must advocate for policies that ensure access to affordable insulin and quality diabetes care for all. We must empower individuals with diabetes to take an active role in managing their condition, providing them with the education, resources, and support they need to live long, healthy lives. The journey ahead requires collaboration, innovation, and a steadfast commitment to improving the lives of people living with diabetes. Together, we can create a future where diabetes is no longer a barrier to a healthy and fulfilling life.